We are starting the intrinsic pathway initiation phase. When the DNA damage is beyond repair, anti-apoptotic molecule BCL2, BCL1, MCL1, BCLX. I have not written here BCLX. You should write 4 names: BCLX, BCL1, BCL2, and MCL1 are lost. They actually guard this cell, so that the cytochrome C doesn't leak out and there will be no apoptosis. But if there is DNA damage beyond repair, these anti-apoptotic means they prevent the apoptosis. That's so known as anti-apoptotic molecule. They get replaced by pro-apoptotic molecule. What are the pro-apoptotic molecule? Bax, beam, BAD. Now these molecules are pro-apoptotic, means they induce apoptosis. How? By increasing the mitochondrial permeability. What will happen if increase mitochondrial permeability? There will be leakage of the cytochrome C into the cytoplasm. Generally, it is present inside the inner membrane and outer membrane. Between the inner membrane and outer membrane, it is present. Cytochrome C now gets out into the cytoplasm and activate APAF1 along with procaspase 9. Now procaspase 9 get converted into activate caspase 9. In extrinsic pathway, there is conversion of procaspase 8 and 10 into active 8 and 10 caspase. But in intrinsic pathway, there is conversion of procaspase 9 into activate caspase 9. So the end products of extrinsic pathway initiation phase is active caspase 8 and 10, and the end product of the intrinsic pathway is activate caspase 9. Now execution phase, conversion of both extrinsic and intrinsic pathway will occur. Extrinsic pathway, the result will active caspase 8 and 10, and in intrinsic pathway the result is active caspase 9 formation. Now these two get execute means they merge together and activate caspase 3, 6, 7. This in turn sequentially activate all other caspase, that will lead to caspase cleave. Now all the caspase are activated and the caspase will cleave cytoskeletal and nuclear matrix protein. All the caspase, they will now cleave the cytoskeletal and nuclear matrix protein, that will lead to cellular degeneration and apoptotic bodies formation. These apoptotic bodies now get phagocytosed and apoptosis will occur. What is efferocytosis? It's an important term, efferocytosis. This means the immunological silent removal of apoptotic cell by efferocyte. What are efferocyte? They are only phagocyte. This phagocyte or efferocyte, they immunologically silently remove the apoptotic cell. In healthy cell, there is presence of CD47 receptor on this cell that display don't eat me signal. That's why they don't get phagocytosed by the phagocytosis. Why the phagocytes don't eat the healthy cell? Because this hit the healthy cell, so CD47 receptor, this is the receptor which display don't eat me. But in apoptotic cell, there is a phosphatidylserine that is generally present on the inner membrane. Now it has flip-flop, means inner get towards the outer. Now phosphatidylserine which present on the inner side of the cell now get exposed in the outer side. That display eat me signal. That is externalization of phosphatidylserine. Now phagocyte engulf it. What are the morphologic changes in apoptosis? Now the cell will decrease in size. I have made the diagram a little wrong. I should make this one the large and this one the small. You should make this one large and this one small, because there will be cellular shrinkage. First the cell will shrink. Then there will be increase in eosinophil. That means eosinophilia will occur. ... are eosinophilia. Now it leads to pycnosis, means compaction of the nuclei, and then it leads to karyohexis, means fragmentation of nuclei, and there will be blebs, means irregular cellular margin or bleb formation. Then there will be apoptotic bodies which get phagocytosed by phagocytes and apoptosis will occur. How will diagnose the apoptosis? Diagnosis will occur by 2 points. We'll write apoptosis marker. What is the apoptosis marker? It's the annexin 5. Annexin 5 is a recombinant protein with higher affinity towards phosphatidylserine. There will be flip-flop. There will be externalization of phosphatidylserine to which annexin 5 will bind, and we can see. And now this phosphatidylserine annexin 5 bind to phosphatidylserine and act as a marker of apoptosis. Second is agarose gel electrophoresis. If we check the cell in electrophoresis, there will be fragmented DNA. So the step ladder pattern due to endonuclease activity. Endonuclease break the DNA into step ladder pattern. Thank you.